Hello, everybody. Let's gradually start. Stephanie, are you with us?
I am, thank you.
I don't hear you yet. Okay, now.
Can you hear me?
Yes.
Excellent.
Okay, very good. So welcome everybody for the talk of Stephanie Jones. Just a few words about Stephanie. So Stephanie is a mathematician by training, and she did her bachelor and master degree in Boston College. She back then specialized in dynamical system theory and computational neuroscience, and her supervisor was Nancy Capel, whom of course many of you know very well. She then did a lot of training in MEG and EEG in Martinez Center for Biomedical Imaging, and then she became an assistant professor, and now she is an associate professor in Brown University. So now Stephanie's research integrates human brain imaging and computational neuroscience methods to study brain dynamics in health and disease. So Stephanie works closely with animal neurophysiologists and clinicians to develop data which kind of allows her to constrain neural models that are functionally and importantly clinically relevant. So now, of course, many of us know Stephanie as the main driving force behind a human neocortical neurosolver software package that is based on biophysically principled neocortical column model that allows simulations of MEG and EEG data. And that's, of course, very important in the context of our research here when we try to understand non-invasive measures of, like based on EEG and MEG. And with that, I welcome Stephanie to present her research.
Thank you. Thank you very much for that kind introduction and for inviting me to be part of this really exciting symposium. I'm so sorry I can't be there in person, but I'm thrilled to have the opportunity to talk about our research program and to get the feedback from this community. So I'll start sharing my screen.
Yes, we see it, Stephanie. Yeah. Okay. But we still don't see you. We don't have your video yet.
Does that look right?
No. Not yet. Not yet.
Let's give it a moment. I am sharing my screen. But what are you seeing?
We see some part of the screen in the left. Like it's your presenter mode, but not like a.
So let me see if I can switch it. So let's see. What if I just do? Maybe I just need to switch.
Mm-hmm.
Does that look right?
Yes, perfect.
Perfect.
Yes.
Thank you. So thank you again. I'm very glad to have this opportunity to present, and I'm going to be talking about our software, Human Neocortical Neurosolver, which is designed for cell and circuit level interpretation of EEG and MEG. And today I'm going to focus on our studies using HNN to understand the mechanism and meaning of brain rhythms in somatosensation. So I'm going to begin by giving you an overview of the challenge in human neuroscience that we aim to address with the development of this tool. So we all know that EEG and MEG are really powerful technologies to study human brain activity because they give us direct correlates of human information processing and neuropathology with millisecond resolution. But a downside is what we're recording is so-called macro scale activity that's generated by large ensembles of neurons, and it's still difficult to infer how these signals are generated at the cell and circuit level. And this circuit level understanding is really critical if we want to use these techniques to understand information processing or to target treatment when they're disrupted in pathology. And so intracranial recording and modulation techniques in animal models are ideal for dissecting circuit level activity, but there's obvious challenges in applying that in the human. And so to bridge this gap, we've been applying computational modeling techniques. And the first type of modeling is current source estimation modeling. This is the type of modeling that takes the extracranial measured signal and estimates the location time course of these big electrical currents in the brain that create the sensor level data. From there, we need to connect these electrical currents to their underlying circuit level generation. And this is the ideal problem for computational neuromodeling, where we can simulate the electrical activities of the neurons, and we can have specificity both at this microcircuit level and at this macro scale recording level. And so this has been the primary focus of our research program, and we've turned this modeling framework into this user-friendly software human neocortical neurosolver, or HNN, that we're distributing to the community and trying to help them develop and test hypotheses on the circuit origin of their data with our software. So in my talk, I'm just going to begin by giving you some of the background on the development of this modeling tool. And then I'm going to give an example of how we've applied it to study the mechanisms and meaning of brain rhythms and somatosensation. And I'm going to be focusing on studies from my lab, where we've looked in particular at these 15 to 29 hertz so-called beta rhythms and their role in tactile detection. And in the very end, I'll briefly describe some other HNN applications to study healthy and pathological cognitive processes. So how do we begin to develop a model that can connect these macro scale signals to the underlying cell and circuit level generation? Well, there are two initial steps to understand in connecting EEG and MEG to circuit level details. The first is that when we apply these inverse solution methods, what we're doing is we're estimating the location, the direction, and the time course of what we call primary electrical currents. These are the big electrical currents that create the recorded sensor data. These primary currents are known to be generated by the postsynaptic intracellular current flow in these long and spatially aligned cortical pyramidal neuron dendrites. Essentially, the length of these and their parallel alignment creates a net big electrical current that creates an electrical and a magnetic field that can be recorded outside of the head with our EEG and MEG. So given that this is where the signal comes from, there are some key canonical cortical circuitry features that we should take into account in our model if we want to connect to the circuit level detail with our EEG or MEG signals. The first is that the cortex is a layered structure. It contains synaptically coupled excitatory and inhibitory neurons across the layers, including these pyramidal neurons with these long apical dendrites in both the supragranular and infragranular layers. These networks don't sit in isolation, but they're constantly receiving input from other parts of the brain. And there are two primary pathways of information flow into these cortical circuits. One pathway is from the lemniscal thalamus. This is the pathway that relays sensory information through the body, up through the brain, to the thalamus, and into the cortex. In the model, we refer to this as proximal drive because it effectively targets the proximal dendrites of these pyramidal neurons. The other pathway of information flow comes right into the supragranular layers. This is information from higher order cortical circuits or non-lemniscal thalamic nuclei that project right up to the supragranular layers. And in the model, we refer to this as distal drive because it effectively hits the distal dendrites of these pyramidal neurons. And again, this big primary electrical current comes from the net intracellular current flow across the whole population of pyramidal neurons. And with this construction in the model, the units of measure that we get out are current over distance, or nanoamp meters. This is the same unit of measure that we get from our source localized data. And so we have one-to-one comparison between the output of our model and our source localized signal, which turns out to be a very powerful way to start to study the circuit origin of these signals. So this is a reduced representation of the model in HNN. Oh, first I want to describe, in HNN, we're not actually simulating all of these brain circuits that activate these local cortical columns. Instead, what we do is we generate trains of action potentials in predefined patterns. And these action potentials activate excitatory synapses in the local network that activates local network activity and drives current flow up and down these pyramidal neuron dendrites. And so this is actually a reduced representation of the model. The full model contains a scalable number of neurons, and it represents a patch of neocortex in a localized brain area. And we've been specifically designing the software to study some of the most commonly measured signals from a localized brain area, including event-related potentials and low-frequency brain rhythms. Now, this is a really large-scale complex neural model. It's open source. It's developed in the software Neuron Python, and it's openly distributed, so all of the code is available. But unless you have some experience building and working with these large-scale neural models, it can be really daunting to work with this underlying code. And so to make this accessible to a broad MEG and EEG community, we've embedded the model in a graphical user interface. We created a website that contains installation instructions for all platforms, as well as tutorials on how to study event-related potentials, alpha rhythms, beta rhythms, and gamma rhythms. We describe these tutorials by giving you example data sets and parameter sets from our prior published work, with the idea if we can teach you how we did it, then you can use the software to study your own data and test your own hypotheses. HNN also has a Python interface, and we're working really hard to construct it with best practices in open source software design so that we can engage the community in its development and applications. And in my talk, I'll be showing you some practical examples of how this HNN graphical user interface can be used. So just to summarize the background, HNN is a biophysically principled neocortical model. It's embedded in a graphical user interface, and it's openly distributed with tutorials of use for ERPs and low frequency brain rhythms. So how did we use HNN to study the mechanisms and meaning of these beta rhythms in somatosensory perception? So in my lab, we've been studying somatosensory perception, and we perform tactile detection experiments where we give the subject a brief tap to the finger and we ask them, did you feel it? Did you not feel it? These are very boring tasks, but they give very reliable and beautiful EEG and MEG signals. And what I'm showing you here is MEG data, but we see something very similar in our EEG data. And our main interest has been on understanding signals from the primary somatosensory cortex. And so we've applied a standard inverse solution technique here to isolate the contribution of the signal from the hand area of S1. And I'm showing you the spontaneous signal from the hand area of S1 before the tap to the finger occurs. And what you can see, there's a large amplitude, low frequency oscillation, and we put a frequency filter on it. This is just a standard wavelet analysis. You can see there's a strong component in this 15 to 29 hertz beta band. But the beta expression changes at a moment by moment instant in time. And so the question we were really interested in was, does the power of this beta in the pre-stimulus period predict whether the subject will feel that brief tap to the finger or not? And it turns out it's a very reliable predictor of perception. And so what I'm showing you here is we've taken this pre-stimulus beta power, we've averaged across the 1 second pre-stimulus time window, we've averaged across the frequency band, and we've sorted power from low power to high power. And on the y-axis is the detection rate as a percent change in hit rate from the mean. And so the lower it is, the more likely you are to perceive. And so with high pre-stimulus beta power, we have inhibited perception. And so in general, we think of beta power as being a signature of inhibited perception. We've also observed that these beta rhythms have a direct impact on the processing of that incoming sensory information. And so what I'm showing you here is the evoked response from the hand area of S1, where we've sorted over trials where the subject reported detecting or feeling that tap to the finger. On the left here in blue, this dark blue line. And on the right, I'm showing you the evoked response when subjects reported not detecting the tap to the finger in dark red. Overlaid are the responses instead sorting over high and low pre-stimulus beta power. And what we see is that when we have high pre-stimulus beta power, the evoked response starts lower, we get a wider peak and a shallower rise. And this is very consistent with what we see on non-detected trials. And similarly, for sorting over low pre-stimulus beta power in detected trials, which suggests that there's this causal relationship between beta rhythms and the evoked response that has a direct relationship to the inhibition of detection. We've seen several other pieces of evidence that suggest that beta rhythms are inhibiting processing. We've also done these studies where we cue the subject, pay attention to your finger or your foot. And we find that when we say pay attention, we look in the hand area, When we say pay attention to the hand, these beta rhythms decrease in the post-cue pre-stimulus period. This decrease is in coordination with circuits in the frontal cortex, and in particular, the right inferior frontal junction, which is known to mediate top-down attentional control via the inhibition of non-relevant representations or motor responses. And so what these results are suggesting is that beta rhythms are getting out of the way with attention in order to have optimal information processing in that hand representation. In one of our most recent studies, we've shown that this beta decrease with attention can be enhanced with mind-body meditative practice. And this is a study that was spearheaded by my dear friend and colleague, the late Dr. Catherine Kerr. Kathy unfortunately passed away several years ago from a long-term cancer illness. And Kathy practiced the mind-body intervention known as Qigong, which incorporates gentle, rhythmic, and repetitive movements with breath and meditative practice. And it has a rich history in Chinese medicine. And Kathy believed that this Qigong helped her survive with her cancer for many years. And with some donor support, she was able to run a study called the Vitality Project that compared Qigong and exercise nutrition, in this case, Pilates and plant-based nutrition, on measures of fatigue, emotional health, and stress in female cancer survivors. And these were measured with questionnaires. And we also were able to assess brain-body changes, including the assessment of mechanisms of attentional regulation with EEG. And so this task was similar to our attend hand, attend foot task, but instead subjects were cued to attend to either sensation in their right or left hand. And we were using EEG and looking at sensors above the sensory motor cortex. And here I'm showing you the average data above both hemispheres. And what you see is a similar phenomena where the subject is cued to attend to the hand, there's a decrease in beta power. This decrease appears to be more effective in the Qigong group. That suggests that they may be using these beta rhythms more effectively to regulate attention to the body. These EEG results are not yet published, but what we did just publish were the results from our primary outcome measures, which was our questionnaires assessing fatigue. And what we found was that in both groups, fatigue was improved after 10 weeks of either intervention practice. And this is a really important outcome because Qigong is a much gentler intervention and may be more attainable than exercise and nutrition for these cancer survivors to regulate their fatigue. We also found that Qigong improved mood, emotional regulation, and stress, while exercise and nutrition had a greater impact on improving sleep. And we're currently assessing if any of these measures are related to these changes in the EEG that we're seeing in our attention tasks. We have several other measures where beta is inhibiting processing. We've observed that beta power increases with healthy adult aging. And here we're just looking at between 20 and 45 years old. But there's now more recent studies that show that this is also occurring in older aging from 18 to 80. There are many studies from other groups that show that beta power is overexpressed in Parkinson's disease, and that treatments that alleviate the symptoms of motor inhibition also decrease this overexpressed beta. There's evidence in frontal cortex that beta is an index of inhibited motor action in stop signal tests. So all of this evidence suggests that in general, beta rhythms can be thought of as inhibiting processing. And so the ultimate question we're interested in is why? What is it about the mechanisms of generation of beta that cause it to have this inhibitory role on sensory and motor function? And so to answer this question, we're going to now turn to our HNN modeling. And I'm going to go back and focus on our tactile detection study. And so recall, the more beta you have in the pre-stimulus period, the less likely you are to feel that tap to the finger. And this beta power has a direct influence on the processing of that sensory information, such that the more beta power you have, you start with this lower evoked response, and then you have this wider and shallower peak. So the question is, can HNN help us understand why? So to address that question, the first thing we're going to do is we're going to apply HNN to study the mechanisms of generation of the beta rhythm. Then we're going to apply HNN to study the mechanisms of generation of the ERP. And then we're going to put these two mechanisms together and see what HNN predicts about what's going on in the underlying circuits. And so starting with applying HNN to study the mechanisms of generation of beta. The first thing we did was we went back and we looked more closely at beta rhythms in unaveraged data. The brain isn't averaging. Everything I've shown you so far is averaged over multiple trials, averaged over the frequency band, averaged over subjects sometimes. But what we started to do was unpack, what does this look like in unaveraged data? So here I'm showing you a single example of 1 second of pre-stimulus data. Here's the raw time series, and here's the frequency spectrum analysis. And what we found was that these beta rhythms actually occur very transiently. We've taken a closer look at the waveform shape of this beta rhythm. And so we go in and we find the high power activity, and we go back and we look at the raw waveform. And what we see is that there's a very stereotypical shape underlying these high power beta events. And here I'm just showing you 50 high power beta events overlaid, and this is the mean and average across subjects. We quantified many things, but I think it's very clear that these are dominated by this negative deflecting peak that lasts 50 milliseconds. And because this peak lasts 50 milliseconds, when you put a frequency filter on it, you get high power in the beta band. Now, this transient expression of beta and this stereotyped waveform shape has now been seen in many studies. It's seen in sensory cortex, it's seen in motor cortex, it's seen in frontal cortex, which suggests that there's some very robust underlying phenomena creating this waveform shape of these beta events. And so we're going to apply HNN to try to understand, how is this waveform shape generated? So how do we use this very large scale cortical column model to study the mechanisms of generation of this waveform shape? Well, there were several steps that we took in order to constrain this question. The first thing that we did in generating this large scale model is we developed the model of the individual cells in their morphology. And we spent a lot of time tuning the features of the cells to have realistic spiking patterns to injected current. And then we fixed all of those parameters. We then built in the local connectivity structure. And again, this is based on a large body of literature from animals that suggests how these cortical circuits should be connected within and across the layers. We tuned all that, and then we fixed it. And the only parameters that we're playing with in order to understand the generation of this waveform shape are the timing and the strength of these exogenous inputs that come in and activate the local network through these proximal and distal projection patterns. And through a long history of simulation and trial and error and hypothesis testing that I don't have time to get into the details of, the punchline of what we came to was that these beta events are generated from the integration of a broad proximal drive that's simultaneous with a strong distal drive that lasts 50 milliseconds. And so what I'm showing you here is a histogram of the spikes that are going to activate this local network. This is the broad proximal drive, and the distal drive that's stronger and lasts approximately 50 milliseconds. And so what happens in the model with this type of input pattern is this proximal drive comes in and it drives current flow up the pyramidal neuron dendrites. And if that's all we had, we'd have this single peak that goes up like that. But at the same time, we have this strong distal drive. This distal drive is pushing current flow down the dendrites and breaking that upward current flow, and it lasts 50 milliseconds. And so this was a purely model-derived prediction, novel prediction on the origin of these beta events that really came from just trying very hard to match the output of the model with our data. And due to the level of biophysical detail in the model, we could then go in and test these predictions. The first thing that we see is it does very reliably reproduce features of our MEG data and also very similarly EEG data. And then we were able to validate some of these predictions with laminar current source density recordings in both mice and monkeys, and more recently with laminar high resolution MEG in humans. And so we have some support that this beta mechanism is occurring in across species and across recording modalities. Okay, so can HNN now help us understand how these ERPs are generated? And then we'll put together this beta mechanism generation and our EEP and see what it says. And so how do we go about studying the mechanisms of generation of the ERP? Well, actually, the ERP was the first thing that we studied with the model. And this was our first study before it was HNN, when it was just a model on my computer. And what we wanted to do was see if we could test some theories about where somatosensory evoked potentials comes from. And there's a long history of research on the origin of somatosensory evoked potentials. And so here I'm going to show you how we go about doing this in the graphical user interface in the software. So the first thing that you can do is you can load in your data. And this is the data from a strong tap to the fingertip, source localized to S1. And then we can test some theories on how is this ERP generated. And again, there's a long history of literature in animal models looking at the generation of this ERP. And what it suggests is that the ERP is generated by a sequence of activation of this local S1 network that consists of a feed-forward input, approximately 25 milliseconds after you give the sensory stimulation, followed by a feedback or distal input in our model at about 70 milliseconds, followed by a re-emergent feed-forward input that comes about as part of a thalamocortical loop at approximately 125 milliseconds. So in HNN, we can test the hypothesis that this sequence of activation can generate this recorded macro scale ERP. And so what we did was we built in this sequence of activation. So this is a histogram of the proximal and distal drive patterns that we're going to activate this local network with. When we hit run simulation, these inputs come in, they activate excitatory synapses, they drive spiking activity in inter-cell interactions in the local network, and then we can record the macro scale signal. And what you can see is that with this input pattern, we can very nicely reproduce this ERP. Now, importantly, the literature gave us some guidelines as to where to begin, but we had to spend a lot of time tuning the number of inputs, their strength, their timing, in order to get a good representation of this signal. And we have some parameter optimization tools, HNN, to help you do that. And now we could also go in and look at what's going on in the cellular level activity, what's going on in the different layers and the different spike patterns. I'm not going to have time to go into the detail of all that now, but that is all possible to be visualized in our HNN software. Okay, so we have our mechanism of generation of these beta events. It comes from the integration of these two inputs, where the main thing is this strong distal drive. We have our generation of an ERP, which comes from a sequence of information flow into the local cortical circuit, feed forward, feedback, feed forward. Now we're going to put those together. And what happens in the model when we put those together is the following. We are very nicely able to reproduce what we're seeing in our MEG data. And so what we're doing here is we're simulating a pre-stimulus beta event or no pre-stimulus beta event. And I meant to mention to you that high pre-stimulus beta power corresponds to one or more beta events, where low pre-stimulus beta power corresponds to no events. And so if we simulate in our model a beta event with a tactile evoked response, then we get this blue ERP out of our, red ERP out of our model. If we don't have a beta event and we simulate an evoked response, we get this blue ERP. Very nicely reproduces what we're seeing in our data. And I always like to stop at this point in the talk and point out that we actually did this in the model before we looked for it in our data. In our model, we had a prediction of beta events. We had a prediction of ERPs. We said, what does the model predict if we put these together? And this is what it predicted. And then we went back and we looked for our data, and this is what we found, which was really quite remarkable and suggests that once you have the right level of detail in the model, it can be generative of new and unexpected predictions. Okay, so what's going on in the underlying circuit? Why is it that when you have a beta event, you get an evoked response that looks like this, and it corresponds to, I didn't feel the tap to the finger. Or if you don't have a beta event, you get a larger evoked response and the subject says, I did feel the tap to the finger. Well, what the model suggests is as following. Remember, beta is created by this strong distal drive. This distal drive is not only hitting the distal pyramidal neuron dendrites, but it's also recruiting a lot of inhibition in the supragranular layers. If you have a lot of inhibition in the pre-stimulus period before you tap the finger, then what happens when you tap the finger and this sequence of feed forward and feedback information flow come in, it gets shunted. And so this feedback input can't get through. And so this current down is weaker and you get less spiking in your pyramidal neuron populations. And so you get a smaller amplitude and shallower rise in this late period. And so if you have a decreased spiking in these pyramidal neurons, then you have a decreased relay of upstream information out of S1. And if you can't get out of the hand area of S1, then the brain says, I didn't feel the tap to the finger. And so you'll have a decreased probability of perception. So just to repeat that, beta rhythms are recruiting inhibition in the supragranular layers. When you give a tap to the finger, this feedback input can't get through, so you get less spiking in the pyramidal neurons. That decreases the amplitude of this peak, decreases the relay information out of S1, and the subject says, I didn't feel the tap to the finger. Now, this is a model-based prediction that we're currently testing in collaboration with invasive recordings with collaboration with my colleague, Chris Moore at Brown University. And so this is just a slide showing a little bit more detail in the underlying model. What I'm showing you here is the spiking activity in each of the populations of neurons, the pyramidal neurons and interneurons in layer 2, 3, and layer 5 in the model. And here is the time where we tap the finger. And our simulated tap to the finger, remember, is an input at 25, 70, and 135 milliseconds. If we have a beta event, we get a lot of pre-stimulus inhibition so that when that tap to the finger is simulated, it can't get through and we don't get any spiking in the population. Where if we don't have that inhibition, we can get very robust, reliable spiking. The signal is relayed out of S1 and the subject feels the tap to the finger. Okay, so to summarize, beta events emerge from a broad proximal drive that's simultaneous with the distal drive that lasts approximately one beta period of 50 milliseconds. Decreased beta, the model is predicting that that beta decreases the salience of sensory input via the recruitment of supragranular inhibition, and that this decreases the relay of information out of S1. Okay, so in the last few minutes, I just want to show you some other ways that HNN is being applied to study healthy and pathological cognitive processes. So I've been showing you our results from beta rhythms, but HNN has also been applied to study alpha rhythms and gamma rhythms, and we have tutorials on these as well in our software in several prior publications. We've used HNN to look at the impact of non-invasive brain stimulation on EEG signals in perception. We have a published study looking at the impact of transcranial alternating current stimulation on changes in ERP. And currently in the lab, we're looking at changes in EEG signals with TMS and how they may be impacting tactile perception. With some collaborative work, we've looked at auditory processing. We've looked at changes with healthy aging. Currently, we're looking at changes with Alzheimer's disease. Other groups have applied the software to look at changes in somatosensory rhythms in children with autism. We have several ongoing studies where we're looking at changes in EEG signals with depression, in particular the effects of ketamine for improvements in depression symptoms. We have several collaborations where we're looking at changes in evoked EEG signatures with development, both healthy and abnormal development. And we're also looking at the impact of pain on EEG signals. We have a preprint looking at laser evoked pain, and we have some ongoing work with Friederike Pechner and a shared graduate, Chloe Zimmerman, where we're looking at interoceptive processing and the relationship between the heart and the EEG signal and what it means for chronic pain patients. You can see this full list of publications using HNN on our website, and the methods paper is here in eLife. So with that, I'm going to conclude, and I've shown you that HNN is a neocortical column model that's under thalamocortical and cortical drive. It simulates MEG and EEG primary currents based on their biophysical origin. It's been developed into a software that's distributed with a graphical user interface and tutorials of use. HNN combined with MEG and invasive animal recordings revealed this novel information about the mechanisms and meaning of neocortical beta rhythms. We see that these beta rhythms are actually very transient with a stereotypical waveform shape. We refer to them as beta events. Other groups refer to them as beta bursts, but I think in general, we're talking about similar things. Beta events are emerging from this broad proximal drive that's simultaneous with the distal drive that lasts 50 milliseconds. And beta is decreasing the salience of the sensory input via the recruitment of supergranular inhibition that ultimately decreases the relay of the information out of S1. And hopefully I've convinced you that HNN can be applied to uncover circuit mechanisms underlying both MEG and EEG correlates of healthy and neuropathological brain states. Ultimately, we're hoping that it's going to aid in the development of new treatment strategies, either through pharmacology or through non-invasive brain stimulation. And so with that, I'd like to thank you all for listening and thank my collaborators and funding sources. I have a really amazing lab at Brown, and everybody's contributed to this work in a meaningful way. I've also been really fortunate to develop a lot of amazing collaborations that have allowed us to do this interdisciplinary science. And so what I've shown you today is really the efforts of a large team of people. And so I want to thank everyone for their contributions. And with that, I'm going to end with my conclusions and see if there are any questions.
Okay, thank you, Stephanie. Very beautiful and mechanistic. So now time for questions, comments. We monitor both online and the audience. Arno, yeah.
So you show very nice data on beta rhythm and influencing somatosensory perception. What about alpha? You know that many groups, including ours, have shown indication that alpha also determines sensory perception. Now, how do you see the respective roles of the two? Is there some differential contribution? Are they complementary? Do they differentially relate to perception?
Yeah, thank you for that question. It's hard to talk about everything in one talk. And so here we really do focus on beta and some of the novelty that's coming out of our research. But yes, the alpha also obviously has a very strong impact on perception. And we have seen that in our studies. And we have investigated with HNN. In our studies, what we seem to be finding is that the attentional cue is strongly influencing alpha. Right after we give that attentional cue, you see a shift in the alpha rhythm. The change in beta seems to be more closely tied to perception. Subjects learn very quickly in this task that they have at least a second till they have to process this sensory information. You know, we give the cue, we wait at least a second, and then we tap the finger. And it's not until right before we tap the finger that this beta modulation seems to be coming online. We've also seen that beta has a really linear impact on perception, where alpha seems to have this inverted U relationship, such that if you have a lot of alpha, you're not going to feel, but if you have a little alpha, you may be, if you have an intermediate level of alpha, you may be more apt to feel. So there are some differences in what we're seeing in our data between how alpha and beta influence perception. In our model, we've modeled alpha and beta. And in S1, you get this complex alpha and beta rhythm. Sometimes you see alpha, sometimes you see beta. And what we found in our model is that we're giving these inputs. If we give them rhythmically at 10 hertz, right? We have these thalamocortical drives that come in and they hit the proximal and dendrites of these pyramidal neurons. We give them at 10 hertz. When they come in together and the supragranular is stronger, you get beta. If they come in slightly off, then you're getting alpha. And so what we think is that the thalamus is bursting at this alpha frequency, and there's a mechanism in the thalamus that may be regulating the timing of these bursts to either give an alpha rhythm or a beta rhythm. How and when and why is an open question, but we are starting to use our model and our data to try to tease this apart. And is it that alpha is also recruiting inhibition, but perhaps in a different way? The ERP differences look slightly different with if we sort over alpha than beta. And so we can start to use our model to say, what is the difference if we just look at alpha versus beta, the relationship between the two. So there is a lot of ongoing work in that direction, but right now what the model is predicting is that the thalamus is bursting at alpha. If they come in together and the distal is strong, you get beta. If they come in slightly off in timing, you get alpha. And we have tutorials on our HNN website on how to simulate that based on the published studies that we have on this. But to answer the exact question of how and why do they differentially impact perception and shift with attention, that's ongoing. I don't know the answer to that, but it's exciting to think about.
Thank you very much.
Okay, so there were almost 3 simultaneous hands going up. And I hope our beta wasn't too strong with Paul, so we detected the order in the following way. So it's Tilman, then Patrick, and then Karina.
Hi, thank you for the very fascinating talk. Actually, my question was in a similar direction as the previous one, but maybe a follow-up question to that. So you were mentioning cortico-cortical beta effects and thalamocortical beta effects, and now I wondered, the distal drive versus the proximal drive. I mean, I guess proximal drive would be thalamocortical and distal one cortico-cortical, or do you have assumptions about that as well?
Yeah, that's another really important question. So in the model, it's agnostic, right? All we know in the model is that it comes in here or here. But then we have to think about, well, what's a logical place for those to come from? And what we're hypothesizing right now is that in these spontaneous pre-stimulus states, before we tap the finger, these beta rhythms, we are hypothesizing, are coming from 2 different thalamic nuclei. They may also be coming from a thalamic nuclei, the posterior medial thalamus, that projects right up to both the supragranular and infragranular layers. Or it could be lemniscal and non-lemniscal thalamic nuclei that have more dense projections, either proximal or distal. And we're trying right now with our collaborators to test this a little bit further and look at activity in the thalamus simultaneous with the cortex to see, is this in fact the case? It could be that there are intracortical connections from motor cortex that are also contributing. This is an open question at this point, but we are hypothesizing based on prior literature in the anatomy of the circuits that it may be coming from thalamic sources. Now, when you tap something or you do something and then you see beta, that may be driven, may have more of a propensity to be driven by intracortical sources, but that's not what we're looking at the moment.
Cool. Thank you very much.
Okay, I wasn't sure if there was another part of that question.
Okay, now, yes, Alina, thank you.
Thank you very much for this really interesting and detailed talk. So my question is also about the distal drive. So you said that the distal drive lasts 50 milliseconds. It's the basis of the time constant that we see in beta. And the question is why? You know, why 50 milliseconds? Why does it come in packages of that duration? Do you think this is because of the time constant of the local inhibitory network within superficial layers of S1? Or do you think it's to do with the pulsatile nature of the inputs from either other cortical areas or the thalamus? So there must be some profound reason why it's...
Yeah, it's an excellent question, and one we've thought a lot about. You know, and there's some evidence that shifts in attention may be happening at a beta frequency. Some of the work by Earl Miller and Tim Bushman suggests that, you know, that attention comes in these beta packets. But at the mechanistic level, in the model right now, the way that we're getting 50 milliseconds is by creating that burst to last 50 milliseconds, right? We're giving a train of inputs, and I don't know if you can see my arrow right now, but we give this input over 50 milliseconds. There's other ways that 50 millisecond time constant could come about. It could be that the synapses are activated for 50 milliseconds. It could be, one of the things we're thinking right now is it could be calcium mediated in the fact that these are driving dendritic calcium spikes. Calcium plateaus tend to last about 50 milliseconds. And so while the initial prediction from our model is that there has to be something excitatory that's happening here for 50 milliseconds, we do think that there are several targeted mechanisms that may be creating that 50 millisecond time constant. And it doesn't have to be that this burst lasts 50 milliseconds. Now, that said, there is evidence in thalamus, if you do whole cell patching from thalamic bursting nuclei, that the burst tends to last about 50 milliseconds, again, because of the calcium dynamics that are creating the burst of activity. So it's an open, untested question, but I think there are several targets that we can start to look for to say where is the 50 milliseconds coming from? The other question is why 50 milliseconds? What is it about that 50 millisecond period that's important for cognition? And that's and that may be related to how long this inhibition lasts, maybe related to coordination with upstream circuits. There's a lot of things that could be involved in the why 50 milliseconds question. The mechanisms of 50 millisecond have several targets of where that might be coming from. And right now we're presuming in the model that it's this 50 millisecond bursting in the thalamus. But that could be proven wrong at this point.
All right, so now it will be Karina and then Lyubov in chat.
Do you hear me? Yes. Yeah. Very inspiring talk, super exciting studies. So my question is a bit different. I always wanted to hear your opinion on, I'm sure you're familiar with the work by Bernhard Spitzer, Saskia Hagen's group, where they say that beta is more like related to working memory. So basically you keep something alive and then shortly before something happens, this content gets reawakened. And so I wonder, because in your tasks, I hope I'm not wrong, you usually have a cue and then they get the stimulus, right? Do you also see the beta bursts when there's no cue? And how do you think, like is beta really working memory, or how does it fit in the story of that it changes perception?
Yeah, that's a really good question. And the ultimate answer is, we don't know the answer to that question, but there's some hints that beta may be behaving in a similar manner. And we are collaborating closely with Saskia in some of this work. So yes, we do see that these beta rhythms, we've been focusing in my lab on somatosensory and motor cortex and frontal cortex, not looking at memory per se, but we do see that even without a cue, you can get these spontaneous beta events in the sensory and motor areas. In the memory literature, one thing that may be happening is that indeed beta is coming online during the memory consolidation and the memory encoding and maybe helping with that reinstantiation of the memory. But what we don't know is that beta is blocking the things that you don't want to remember rather than enhancing the things that you do want to remember. I don't know that we have the spatial and temporal resolution, particularly the spatial resolution, to answer that question. Saskia does see, and so does Earl Miller in the literature on this, that these things do occur in bursts, They've also looked a little more closely at the waveform shape, and it's remarkably consistent. I'm not sure if any of that has been, I'm not sure where that's been published, but it is remarkably consistent that these beta rhythms in memory tasks in parietal areas have a very similar waveform shape as to those that we're seeing in sensory and motor cortex, which suggests the mechanisms of generation may be similar. But I don't rule out that the mechanisms of generation in memory may be different than what we're seeing here, but there are some hints that there's some similarities. And maybe one way to reconcile the two is that beta in that context is actually blocking out the things you don't want to remember rather than enhancing the things that you do remember. But it's an excellent open question.
Okay, now we have a question from online community.
So we have a question from Luba Vasileva. Thank you for the great talk. I have a question. How does the HNN model take into account interareal differences when modeling the proximal and distal drives for different cortical areas?
That's a great question. Right now, it doesn't. So right now, HNN just is considered a canonical neocortical column based on what we think are generalizable features, which is layers with pyramidal neurons and supragranular, infragranular layers, these input pathways that tend to be segregated between infragranular and supragranular inputs. How much the finer details of neocortical circuits and input patterns on different areas matter when we're looking at macro scale, we actually don't know the answer to that question. And so we're starting with this canonical structure, but we're trying to develop HNN in a way that it can be expanded so that we can start to include some of these more finite details, different types of inhibitory neurons, different connection pathways between sensory and motor areas, and ask the question, well, how does that change the signal? Does it change the signal in a way that we can measure it outside of the head? You can see differences at the microcircuit in the spiking activity and the calcium flow that are different in the neurons, but create the same ERP. We've seen that in some of our studies. And so how much that finer detail matters when we're recording at the macro scale, we just don't know the answer to that yet. But we're developing the software so that hopefully the community can start to ask that question, can expand the model, can test other hypotheses, can see what's consistent across brain areas as the model details grow. That's a great question.
All right, more questions? Well, while people are thinking about them, I have a few of my own, if you let me. So now I have something relating more to empirical findings. So we know that in human brain there are all kinds of beta. So just I can mention you immediately 3: spontaneous beta, then the ERS beta post-movement, which is relatively high frequency, 25. And then we have corticomuscular beta, which is usually 15, 17 hertz. So my question to you is, if we are talking about spontaneous beta, which is about 20 hertz, unfortunately, it's also a harmonic of alpha. So now, what's your take on this? Do you think there is genuine beta which is locked to alpha, but which is still a genuine beta rather than non-sinusoidal shape of oscillations? And if so, how do you think we can disentangle these two?
Well, the way that we've disentangled it in our data is to look at them over time. And what we see is that beta and alpha aren't overlapping in time with our methods. And so if they were going to be harmonic, then if you see an alpha, you should see a beta. That's not the case in our data. And that's one way I think that when you do your data analysis, you can start to untangle, are these actually harmonic, or, a harmonic is just something that happens with a frequency filter. And so just look at the data and see at this point in time, am I getting both? Is one directly tied to the other? And in our data, that tends not to be the case.
I mean, just to comment on this, on your observation, that's often what we see. But when you do very refined spatial filtering, source modeling, what you actually see is that, of course, we know that there are way more alpha sources than beta sources. So what it means is that there is a special desynchronization of alpha sources that at a macroscopic level, alpha waveforms might look very different from beta. Although if you would be able to have an access more locally to alpha, you would see that the amplitude envelopes of both are following each other. So what I'm saying is that because beta is more local, sometimes alpha cancels out more often than beta because of just phase cancellation on the level of sensors. But in fact, it's a little bit more complex thing. So that's why we are looking now for methods how to disentangle them or how to suppress them in some other way. But you believe that, I mean, do you think that, for instance, a genuine locking of alpha and beta, if it exists, it would also lead to non-sinusoidal shape? I mean, that's also almost philosophical question. It could be real non-sinusoidal shape versus just a locking of genuine oscillations.
Well, one thing is that when you look at the local field potential, when you record inside the brain, and we're presuming you're not getting as much mixing of potentially alpha sources, you see very similar phenomena. And so my intuition is that, yes, if we get our inverse solution methods perfect, right? And so we could actually localize at very fine spatial resolution in our human data. I do think based on some of the invasive animal work that we're going to see that these things are not harmonic. I'm not saying that they're never harmonics. I think for any study, we need to look very closely at our data and really look at our data in non-average, non-post-process, unfiltered ways in order to really get at the answer to your question.
Yeah, very good. Thank you. Oh, yeah.
Yeah, thanks for a great presentation. I wonder whether there is a potential of integrating HNN into source reconstruction algorithms to provide additional constraints so that only the sources or only the components that could arise from real neuronal activity will be reconstructed and spurious ones will be discarded.
That's a fantastic idea. It's something that, this software was developed in collaboration with Matti Hamalainen at MGH, and we're working now with Jyrki and Manak Yas at MGH and other groups to try to think very clearly about, where variability in the model comes from, where variability in this inverse method comes from, how reliable can our estimates be based on that variability. And it's a great idea. It's not something that we're doing at the moment, but it's something that we've talked about quite a bit and would be excited to move forward with.
Okay. Yes, Alina?
Thank you very much. I'm a big fan of your work, and I used HNN. That's a great tool. I recommend everyone. I want to hear your opinion on this waveform shape. So this beta waveform, when you simulate it through HNN, you have a very broad proximal distribution of input and a very narrow distal distribution. And as beta increase, the abundance of beta increase with age, as you show it in the middle-aged participants, I was wondering, what do you think, whether it's the increase of this distribution of the proximal drive that happens due to deterioration of some thalamocortical fibers that happen during aging, or it's actually the narrowing of this distal drive that also happens due to some deterioration.
So we actually looked at this in our 2010 paper where we observed the increase in average beta power with age. At that point, we weren't yet looking at the waveform shape, but what we showed is that the features of the data could be simulated with a more synchronous distal input. And so it wasn't changing the proximal drive, but rather changing the distribution of the distal drive that reproduced features of the data. We now know that these increases with aging, there's a recent study by Tim Barduli and his group looking at the waveform shape with aging, and it is indeed related to features of that downward peak, as well as a little bit those tails. The other thing that happens is you get more of these beta events with aging, and that increase in the rate of events is also contributing to higher power with aging. So I think there's a lot more work to be done in the modeling space with the aging literature, but there is some evidence now of how these beta events are changing with aging. And I encourage you to look at our 2010 paper and also his more recent work looking at this.
Yeah, but maybe just a small follow-up. If I think about it, it's more logical that the fibers between thalamus and corticothalamic fibers are deteriorating. So this like white matter lesions are going on. But then why would this proximal drive why would this distal drive would be narrow with age? What is your guess?
So the narrowness is actually coming from the synchrony of the inputs. And so what it would suggest is that for the distal portion of it, the inputs are coming in more tightly packed in time. How that relates to the integrity of the fibers, I'm not sure. It's a good question, but in our study, it was healthy adults, excuse me, up to the age of 45. And so we were presuming in that study that the tactile detection acuity is actually getting a little bit better. And with that, you're getting a more synchronous, refined input to the supragranular layers, which would create that sharper downward deflection. But I think there's lots of interesting open questions to connect to the aging literature. The other thing that we didn't look at very closely was how is this related to changes in inhibitory tone that occur with aging? And I think that'll be in a really important direction to look at further when we're trying to understand maturation with aging, both healthy and abnormal aging.
Thank you very much.
Yeah, all right. We are just on time. Thank you, Stephanie, again. Thanks a lot.
Thank you all.
All right, so with this, it's we say you goodbye.
Bye. Thanks, everyone. Thanks for the great
Ciao.
questions and for your attention.